So, in last few modules I have covered the processing aspects of different materials, particularly metals, ceramics and polymers, and also I have discussed one of the advanced manufacturing technique, particularly rapid prototyping, and there I have discussed salient features of 3D printing, what is the physics of the drop on demand based 3D printing technique, and some examples of the 3D printed materials. So, in this module and then subsequent modules I will present some case studies essentially highlighting the different aspects of bioceramic development to address how one can enhance fracture toughness of these materials. Now, this is that perennial problem of ceramic based materials, that they are extremely brittle, essentially they have very poor fracture toughness, and while one has to enhance the fracture toughness, that one of the things that one has to take care, that one should not compromise biocompatibility property. So, this is kind of a very key concept in that bioceramics development, that you can enhance the physical properties without compromising biocompatibility properties. So, every time some new compositions or some new materials are developed, while one has to carefully measure the different physical properties, but at the same time ensure that how these new composites would also retain the uncompromised biocompatibility property with respect to some of the conventional or clinically used materials like hydroxyapatite. So, therefore, what I will do actually, I will show you that one of the material science based approach as how to develop that hydroxyapatite titanium composites with better toughness properties without compromising that osteoblast cell functionality in this kind of materials. Just to give you some background as well as to refresh your mind, that what is the typical fracture toughness values of natural bone. So, this is the human cortical bone. People have reported or people have used single edge V-notch beam technique to measure the fracture toughness of the human cortical bone. Now, and they have taken the cadaver bone from different patients of different age, ranging from somewhere between 35 to 90, and what you see here, that this fracture toughness of the human cortical bone varies in the window of somewhere between 4 to 8. So, essentially this window is 4 to 8 MPa square root meter, that is the typical value of the ACVNB long crack fracture toughness of the cortical bone. So, therefore, any material which is to be developed for the intended bone replacement applications, they should have fracture toughness somewhere close to 10 MPa square root meter, because you know that higher the fracture toughness, better is its reliability in certain in their applications like clinical applications. So, this slide also tells you that in the older patients, like patients of the age 70 to 90, if you see that their fracture toughness is much lower compared to the adult healthier patients of lower age group like below 50 and so on. So, therefore, that older patients are more prone to bone fracture during accident, or if they slip it or sleep on the floor and so on and so forth, because their fracture toughness is very low compared to the adult healthy persons. And this is how this single edge V-notch beam technique is used to measure the fracture toughness. So, this is the 4-point flexural configuration. You have introduced a V-notch here, and this notch is introduced in the tensile surface of the or the tensile face of the V-notch samples. So, that the cracks, once this notch is introduced, these cracks will essentially go further, leading to two pieces of the samples. So, bone density is also important in case of the natural bone, and this essentially shows that this fracture toughness scales with the bone density. So, it goes, this is for the cancellous bone, it goes from 0.1 to somewhere above 1 MPa square root meter. Now, bone density, if you remember that bone density essentially depends on the hydroxyapatite content or calcium apatite content in the natural bone. So, higher the relative density of bone means higher is the mineral content. If lower is the mineral content or calcium apatite content, lower is the elastic modulus. So, if you now place this plot with respect to the age of a patient, so this region would be the older patients and this here would be that healthier and younger patients. Okay. Younger patients, because younger patients bone density is relative density is higher, so that fracture toughness value is higher. Older patient bone density is lower, so that calcium apatite content is lower, so density is lower, and then fracture toughness is also low. And this is how the fracture takes place in the cortical bone. As you can see that you have introduced some V notch, then the cracks will go like this, and then this is your osteons, different concentric circles, and then cracks will essentially go through at the periphery of the osteon surface and go towards the other end of the bone structure. Okay. Now, what is the driving force for development of the hydroxyapatite based composites in the bioceramic applications? Essentially, as I said, that most of the hydroxyapatite based materials, if you plot their fracture toughness properties, what you see here, we have tried to summarize here. So, this is that bismuth for B2O5 has been used as a reinforcement, silver has been used as a reinforcement, phosphate glass, zirconia, and various other materials or hydroxide whiskers has been used as a reinforcement. But whatever there is a material, what you see here, except that one single report of the hydroxide zirconia, that all of them are below 1.5 MPa square root meter. So, 1.5 is somewhere between that more higher side of the hydroxide based composites. And then second things that I must mention in this plot, this toughness is measured using indentation toughness. Now, indentation toughness does not give a very reliable measure of the toughness. Indentation toughness means you take a Vickers indent. In brittle materials, these Vickers indents produced some cracks from the fore edge of the corner. Then after that, at any given load, you can measure that what is the crack length 2C1 and this is 2C2, what is the crack length. So, you take average crack length, then based on that you can measure the indentation toughness. So, very briefly, higher is the crack length, lower is the fracture toughness, or in reversely or conversely, lower is the crack length, higher is the fracture toughness. So, physically if I want to explain, this lower crack length essentially means the material is tough enough not to allow crack propagation in that microstructure. If the fracture toughness is high or if the fracture toughness is low, that means material does not have enough resistance against fracture toughness, because remember, fracture toughness essentially means resistance against crack propagation. So, if a material is high fracture toughness, that means material will have higher resistance to crack propagation, and therefore, higher fracture toughness materials you won't be able to see much of the cracks, wherever lower fracture toughness materials you will be clearly seeing this crack generation even at the lower indent load. This drives us to see how to make hydroxide based materials with higher fracture toughness property. So, this is what has been mentioned here. So, what we use is the titanium as a metallic reinforcement. Now, why titanium? Because titanium being a metal, it can enhance the fracture toughness through the mechanism is ductile metal bridging. So, ductile metal bridging essentially is the concept is that suppose you have a titanium particle somewhere here and a crack is going to pass through this kind of microstructure. Now, if this is a titanium particles, this is a titanium particles. So, what will happen? This titanium will undergo plastic flow or plastic deformation at the crack tip, because crack tip has certain stress distribution here. So, this is your titanium, this is your titanium, and once it will ductile flow, then this will flow like this, and if this will flow from both the sides, then what will happen? There is these cracks will this crack tip will be closed because of the flow of the titanium particles on the crack tip. Okay. So, what we do, we have used both the technique, one is conventional sintering like pressureless sintering, and one is the spark plasma sintering. What is conventional sintering? You mix the titanium hydroxyapatite with different proportions of titanium like 5%, 10%, 20%, and then you make the pellet and you throw it inside the furnace like a conventional furnace, like vacuum furnace or argon furnace which is heated in the argon atmosphere. And then at higher temperature you see that whether densification takes place, and then you make the composite. In adverse sintering like spark plasma sintering, which I have mentioned before, like it is a more like passive flowing very large amount of current like 1 to 1.5 kiloampere through this furnace, and then you essentially heat the powder compact by Joule heating, and therefore you can make these composites. Now, there is certain problems associated with the conventional sintering when you particularly sintering air. What you see, that red font phases, these are like different phases which is forming when it is sintered at 1000 degrees Celsius, like what you see calcium titanate, essentially it reacts titanium with calcium oxide and so on, TiO2. Also 200, 1300, 1400 degrees Celsius, you see several other phases are also forming. So, what it shows essentially what it shows is that chemical reactions or sintering reactions taking place because hydroxide titanium both are reactive. So, what we do, we try to sinter them in spark plasma sintering in argon atmosphere at a temperature of 950 degrees Celsius and so on, and then idea is that if you heat them very fast and if you hold them for smaller time period, then you are not allowing the hydroxide titanium to react. So, all the conventional sintering related sintering reactions can be avoided and you can achieve higher densification. The same thing is true here, as you can see that hydroxyapatite is going, this is going to the towards 100% densification. Then when you give titanium and so on, this densification actually is taking place at a much lower temperature, and as a result titanium actually aids in the shrinkage or densification process. Now, as far as the ACM images of this titanium distribution is concerned, what you see, the titanium particles are distributed quite uniformly, but at higher titanium content like 20% titanium and so on, you see that there is a cluster of titanium particles which is distributed in the hydroxyapatite matrix. Now, this hydroxide and titanium, it is both retained very predominantly in 20% titanium and 30% titanium. I think when we published this paper in Actimetry layer, that was the first proof that both hydroxide titanium can be indeed retained in the microstructure, which is otherwise very difficult if one uses the conventional sintering technique. And as you can see, all the characteristic peaks of the hydroxyapatite, all these are lined up at a particular 2 theta angle, which is essentially follows very closely with the JCPDS file of this particular phase. Now, although this any indication of the second phase formation could not be detected within the detection limit of the X-ray diffraction, but we did observe some second phase, particularly the grain boundary region or grain boundary triple pocket here, as you can see grain boundary triple pocket here. Now, this phase, when you did that selected area diffraction pattern, what you see here, it is the CaTi4 PO4 whole 6. That is very unusual phase which does not form during that conventional sintering experiments, but it forms during that spark plasma sintering experiments. And except that, we see that very clear is evidence of the hydroxyapatite and beta titanium and small amount of beta tricalcium phosphate. All these phases are uniquely identified or analyzed using the selected area diffraction pattern from the individual phases. Now, what is the specific reactions that perhaps is taking place in this one? The specific reactions are Ca10PO4 whole 6, that is the stoichiometric HA, and this HA essentially going to beta tricalcium phosphate, what you see Ca3PO4 whole 2 leaving C1H2O, and this Ca3PO4 whole 2 can react with titanium dioxide to form this sintering reaction product, that is CaTi4PO4 whole 6. Why TiO2 comes into picture? All these metals, titanium and so on, they are always covered with a very thin layer of oxides like titanium dioxide and so much. So when calcium phosphate reacts with titanium dioxide, then there is a possibility of CaTi4PO6. It is very difficult to assess the thermodynamic feasibility of this reaction simply because they do not have all these delta G values that thermodynamics gives free energy change of formation of these compounds are not available in literature. Now coming to the mechanical properties of these materials, first of all these materials are sintered more than 95% theoretical density, elastic modulus also is fairly low. It's although little this is that 50 to 60 gigapascal. ACVNB single edge V-notch beam technique, when you measure these fracture toughness values are relatively high, 4.3 MPa square root meter. Remember that earlier one of the earlier slides I have mentioned that which drives the development of the hydroxide based materials, that most of these earlier developed hydroxide based composites, they have a fracture toughness when measured using indentation technique is somewhere around 1.5 MPa square root meter or below, with the exception of one literature case where they have reported close to 3, but that again was measured using the indentation toughness. Okay. Now this is little bit more clear picture about this, how these materials stand with respect to earlier investigated materials. On the right hand panel I have explained that ACVNB technique. So we have used that single edge V-notch beam technique. We introduced the notch here. This notched surface is placed in the tension side of the ACVNB. In this we have used that 4-point bending configuration, flexural configuration, and this is the V-notch here, and then V-notch, and then you measure that K1C values, that is that critical crack tip in stress intensity factor under mode 1 loading, and this is the particular equation one can use. And then when you do these ACVNB measurements here, even the hydroxide itself has fairly high fracture toughness, and then in the 10% titanium one can go very close to 5 MPa square root meter. Remember the cortical bone fracture toughness varies somewhere between 3 to 8 MPa square root meter. So this 5 MPa square root meter, certainly we are almost halfway in the range of that cortical bone, natural cortical bone toughness values. Now as far as the toughness is concerned, what we have observed that when you have titanium on the way, this crack is going, and then crack, there is a evidence of the crack wake debonding and crack bridging, and this is kind of very clearly here you can see titanium here, and around the titanium there is some evidence of the crack bridging as the additional toughening agents. So we have used therefore some of the well known Asby's model or the Evans model just to show that how fracture toughness can be correlated with some of the parameters which can contribute to the fracture toughness in these materials. So these parameters, one of them is S, that is fraction of particle near the crack tip, that is titanium here we are talking about. And second one is the yield strength of the metallic particle, again titanium, because titanium is retained predominantly. D is the particle size, again titanium, and volume fraction of the particles. So what you clearly see that all these things are linearly correlated with delta Kc, because delta K1 is the toughness enhancement because of the presence of the titanium, and they are indeed proportional to square root of each of these term in a linear manner. So higher these values of these parameters, higher will be the fracture toughness enhancement, and that is indeed shown here in our case. Only thing is within the limited data points what we see, it is a kind of clearly linear trend, although they do not follow exactly linear, but they show some increased fracture toughness enhancement of these things. Now this work was published in Acta Materialia in 2013 value. So if you are interested, you can look at this paper for more details. So, this is a kind of a summary of this part plasma sintered materials at 950 degrees Celsius in 30 megapascal and for 5 minutes. It is very indeed very short sintering, and then you do see that evidence of this second phase formation of the grain boundary triple pockets. We have explained that some reactions, that how it is occurring, and apart from that you have a beta titanium, and K1C is a function of percentage titanium. You see that it goes through a very high relatively higher values like 4.8 close to 5 MPa square root meter. Now, cytocompatibility or cell level compatibility of these materials was established using two cell lines. One is the human fetal osteoblast, and it is SAOH2 means sarcoma osteosarcoma, and this is HFOB means human fetal osteoblast. And different assays are done, adhesion, viability and so on. So, first one is that adhesion of SAOH2 cells. So, this is the HA 20% titanium surface on which the cells. Now, certain features you notice that first of all this osteoblast cells, they show more like a flattened morphology, which they should show while adhering on a surface. Second thing, we do observe cellular breach formation like cell to cell contacts, and this is a very characteristic of the osteoblast, and there are some evidence of the extracellular matrix formation on this material, and there is very clear interaction of the phylopodial extension and phylopodial, navelopodia, and then that there is not much significant difference between the control sample and then hydroxylated 20% titanium in terms of that cell morphological changes. And then extension of phylopodial lamellopodia also kind of provide some signatures of the or some signature towards the evidences of the cell migration. This is more evidences of this, how the cell morphological changes has taken place on this particularly SOS2, and then we do see certain evidences of titanium and hydroxyapatite. As expected, cells are adhering more on the hydroxyapatite rich region because hydroxyapatite, as you know, that is that kind of a it is the inorganic composition of the natural bone. Now, this is the fluorescence micrograph, repeating fluorescence micrographs of the SOH2 cells on that hydroxide 5% titanium surfaces, and what you see, this is that actin which is stained in the green, and the nucleus which is stained in the DAPI. Remember that DAPI intercollects with that DNA of the nucleus, and therefore it also gives a blue fluorescence. And then other things that you can see that there is a clear evidences of the cytoskeletal reorganization and cytoskeletal stretching, actin filament stretching on these materials on the cells which have adhered on this material substrate. Now, human fetal osteoblast cells, when they are grown on these materials for 3 days in culture, they do exhibit similar kind of proliferating behavior and forming a very dense cell colony on these materials, and this also active actin filament reorganization in the culture. Now, in terms of the numbers, we have done these experiments for the 2 days, 4 days and 8 days in culture, because these human fetal osteoblast cells are relatively slow growing cells. In all these cases, you see that compared to control, certainly there is a significant compared to, if you consider that hydroxyapatite is one of the control, compared to control that HA 10% and HA 20% titanium, these are significantly higher values even after 8 days of their culture of these cells on these substrates. Now, this work is again published in JBMR B, Journal of Biomedical Research Part B. Now, third aspect is the cell fate processes. Now, these things I have mentioned earlier, that is a cell cycle, cell proliferation, cell apoptosis. All these cell fate processes can be quantified using fluorescent activated cell sorter, which has been discussed before. And in case of this hydroxyapatite 10% titanium, when we have compared with that of the control, what you see here that early apoptotic cells that kind of reduces with the days in number with the days in culture. Similarly, late apoptotic cells also shows certain decreasing trend in terms of time in culture. Percentage necrotic cells and percentage live cells. Live cells also that increases with more time in culture. Now, in terms of cell cycle, we have done some analysis in terms of what is the percentage of cells in G1, S and G2, and what we have observed that G1 percentage G1 it is reducing with more days in culture. That means more cells are going into the next phase, that is the G2, and certainly except with the exception of the control, both on hydroxide and hydroxide 10% titanium, cells are increases, that number of cells in G2 phase increases, and same is true for the S phase, that number of cells are increases. So, what it shows that cells are not stuck while growing them on these substrates, and certainly cells are able to cross the checkpoints while adhering or while growing on these particular substrates. Now, in terms of proliferation, when cells go from first generation, second generation, third generation, and there is a concept called sequential halving. So, therefore, it leads to the generation of distinct peaks, and that is what has been shown here, that it goes from 0 day, and every time it more the time in culture it goes to more on the left hand side. Okay. So, this is that 0 to 8 days, and based on that one can calculate that what is the proliferation index, and interestingly all this number shows that proliferation index systematically increases. That means the substrates, while enhancing the fracture toughness, they indeed actually can be used as a cell growth substrate also. So, this is flow cytometry based analysis that has been again published in a Journal of Biomedical Materials Research Part B. I think with this we have also written a review paper on this hydroxide titanium based composites for bone tissue engineering applications. Those who want to read more about it, they can follow this review paper. And this is the entire summary of this one, that we have sintered this material in 950 degrees Celsius. What I have not shown, that we have also done some biomineralization experiments just to show this apatite crystal formation, which is not influenced because of the presence of titanium. We have shown that how cell fate processes is not compromised, fracture toughness values importantly increased quite some extent, and also early apoptotic cells also decreases because and that shows that these substrates do not induce much of this necrosis or apoptosis.